Hi there. I just wanted to take a few minutes to explain what the Bruno lab does in regards to B cells and tertiary lymphoid structures. Just a little bit of background on my lab. We take a very multi-level approach to understanding B cells and tertiary lymphoid structures in cancer patients. Particularly, we do single-cell RNA-seq, multispectral imaging with paired spatial transcriptomics. We also do small-scale functional assays as well as high-level flow cytometry. If you're looking at this slide, we are often thinking about the pro-tumor, anti-tumor balance of B cells within the tumor microenvironment because there's still not a lot known about these cells. And so our lab is really trying to understand both sides of this coin for B cell function. B cells have been correlated with better prognosis and better responses to immunotherapy, as well as tertiary lymphoid structures. So we like to think that most of the B cells in tumors have an anti-tumor role, which are these potential functions here on the right-hand side. However, there is the potential for inhibitory functions, which have been shown in mouse models, but need to be further refined with a model with tertiary lymphoid structures, which most mouse models do not have. And we are actually generating one of those models as well to back translate some of the key targets we think will be necessary to modulate B cells and tertiary lymphoid structures in cancer patients. I also want to give you an idea of the types of projects that were going on and the types of questions that we have going on. So we can think about how to effectively target these B cells in new immunotherapies and couple them with T cell therapies. In particular, we think about the composition of tertiary lymphoid structures and immature versus mature TLS. And you can read more about this in a News and Views I read about 3 papers that indicate B cells and TLS in immunotherapy response in Nature this past January. And we really think about how do we get to this mature TLS stage? Something that we would see that's very similar in a secondary lymphoid organ. And so these are some of the key questions we're thinking about in the lab. First is, what is the heterogeneity of B cells and TLS in cancer? Second is, how does having TLS influence B and T cell function? And how can we induce these TLS in cancer patients? So we compare multiple solid tumors in order to get at this question. We're utilizing spatial transcriptomics. We're looking at cancer progression, so what happens earlier on before cancer develops, particularly in the setting of lung cancer. We're matching key markers and function together for B cells and tertiary lymphoid structures, and we're interrogating non-immune cells in the tumor microenvironment and how those might influence TLS function. And to give you an idea, here is a spectrum of immunotherapeutic response with the number of somatic mutations in different cancers, and we know pretty clearly that if you look at melanoma, that these patients have really good responses, and these are canonically what we call hot tumors, and they have quite a few tertiary lymphoid structures. However, what we also know is that these middle tumors, such as head and neck and non-small cell lung cancer, also have a lot of TLS. So we work very heavily in non-small cell lung cancer and head and neck, and in particular, we're looking at viral differences in head and neck cancer. So what is different in the HPV positive versus the HPV negative tumor microenvironment? Lastly, we're also thinking about immune cold tumors such as sarcomas, ovarian cancer, and glioblastomas. So there is really a large array of tumor types that you can work on in the laboratory to really evaluate B cells and TLS. We get a lot of fresh tissues, blood, as well as FFPE tissues to do some of our imaging studies. And to that end, since I've been at Pitt, I think we've collected over 700 fresh samples from different tumor indices to do our analyses. We are really looking to back translate, as I mentioned, a lot of these findings into a physiologically relevant mouse model, which is now being worked up and is working quite well. So there is the possibility to do combination human and mouse studies within the laboratory. So with that, I'm going to stop screen sharing and just mention a few other things. I'm really looking for students to come in and captain some of these key questions. I have a really great working group now. I have a couple graduate students. I have a postdoc. I have a couple technicians to help out. So we have a really good working group to get some of these studies done. From a personal perspective, I really enjoy Pittsburgh. So if you have any questions about that, I'm a Pennsylvania native. I was originally born in Erie, Pennsylvania, which is 2 hours north of Pittsburgh. So if you want to know anything about the area and pluses and minuses of living in Pittsburgh, I'm a good person to talk to because I wasn't overly convinced when I moved here, but I've really enjoyed it. I really would welcome the opportunity to get to know any of you. Please feel free to email me at my Pitt email address to learn more about the research program. And I really look forward to having you come into the program this upcoming year and learning more about you. Thank you so much. Bye-bye.